Hello, everybody.
Hello, everybody.
I'm the last one. And it's remind me the movie, the name is The Last Man Standing. So I hope you guys are not tired and will listen my some key findings, what I did during my PhD. And as you know, my name is Waleed Asghar. I got my PhD from Japan. And during my PhD, mostly I worked with the soil microorganisms for the betterment of soil health and the promotion of plant growth. So, as we all aware, soil health is very important. And if we think about the soil health, and we cannot improve the soil health within one day and within 1 year, within 2 years. So we have to work on the long-term process. So being a researcher, how I can do and with my knowledge, with my expertise, how I can improve soil health. So today I will talk about the one of my experiment. So most of the experiment during my PhD, it was a COVID time. So we conducted lab-based studies and incubation studies. So I will talk about the co-application of Trichoderma species with organic compost and the promotion of plant growth and the enzymes activities and the fungal community composition. Trichoderma basically is kind of soil microorganisms as always existing in the rhizosphere and endophytes as well. It can be extracted from the plants, roots, and rhizosphere as well. So we move to the further slides. As we know, the world population is increasing day by day. We can see in the map. So we we can see in the map the world population is increasing in the couple of years, the couple of decades. The population will reach more than 10 billion. So if we think about the population is increasing, then definitely we need more crops, we need more food. So as we see the people are eating food, including me. So but the problem is the land is not increasing, but the population is increasing. So it's a pressure on the farmers to produce more crops. They are using heavily fertilizers to produce more crop to meeting the food demand. But ultimately, they are unaware about how they are going to be disturb the soil health and how they will degrade the land. So, due to the recent published studies, we can see that most of the land is already very degraded, and some of the soils are degraded, but only few soil is remaining as a stable soil. Uh, because some of the farmers are already aware, they use some kind of organic fertilization, green manuring, cover cropping. They try to protect the soil compared to the synthetic chemical fertilizers. So the problem is how we can protect our soil. As you know, you are the great researchers. You know that there are many strategies we can improve the soil. But here I will talk about some strategies like organic manuring, green manuring, mulching, organic composting, and biofertilizers to improve the soil health. So soil is kind of very complex ecosystem and integrated management that provides many benefits to the soil to construct the soil better. Soil health is very important, as we are already aware, as discussed in the previous slides. There are many types of indications, the functions we can improve the soil health. We can know that our soil health is good, our soil health is bad. So, according to the soil health, we have to focus on the soil functions. If we talk about the soil functions, which is the indicator like improve the soil pH, improve the microbial biomass, and improve the soil aggregation, rebuild the rhizosphere by using organic composting, by using green manuring, by covering the cropping. Soil is kind of our natural resources. Soil is a living and the life-giving material or resources. Soil almost have more than 10 billions of microorganisms, including bacteria and fungi, who are the main driven in the ecosystem. Because microbes are always responsible for the nutrient cycling, microbes are always responsible for the aggregation. So that's why here we choose some things to improve the soil health by organic and by biofertilizers. If we talk about the soil health, how we can improve the soil health by organic farming, by mulching and green manuring and biofertilizers. So further we move about the organic composting. So because we have to counter the problems. So organic composting is very famous these days. Organic composting increasing day by day, but as we aware about the increase the organic compost can increase the microbial community. They can store more carbon into the soil. They can improve soil structure, and they can improve the soil structure and root development and reduction of soil diseases as well. But at the same time, we know when we use organic composting as a fertilizer, as not just a kind of substrate, they cannot provide the nutrients to the plants. Most of the nutrients they stuck into the soil, they cannot be degraded and not available for the plants. So we there is another problem, and because we have to improve the soil at the same time, but we have to enhance the plant growth promotion at the same time, because we have to feed the population. That population is increasing, so definitely we need to more food. So there is another problem. So countering this problem, we move to the another strategy. Maybe it can work. So we talk about microorganisms. So we dialogues, we will go to the we will dig the soil to dialogues with the soil microorganism. As we know, soil microorganisms is kind of very sensitive indicators of the soil health. Whenever we want to know the soil health, we can check the soil microbial biomass, how much microorganisms already exist in the soil. So we can predict the early indication of the soil. So if the microorganisms, I mean microbial biomass in the soil is very less, so we must need to promote the microbial biomass by using organic farming, by green manuring, by mulching. So soil microorganisms are always responsible for the soil nutrient supply, organic matter decompositions, and biocontrol diseases, and nutrient cycling and phytohormones production. So there are many microorganisms are already identified from the rhizosphere soil and from the plant roots like endophytes. So they are gaining interest day by day. But most of the macroorganisms are not stable. I mean, sometimes they give very beneficial results, but some of the time they cannot give the better results due to the environmental stresses and other stresses as well. But these days, Trichoderma is gaining more knowledge, more and more attention compared to the other macroorganisms. Trichoderma, firstly, it was introduced as a biocontrol agent to control the some soil-borne diseases, especially occurring in the soil and plant as well, but is now is using as a supplement to promote the plant growth promoter. So our strategy, our hypothesis was created, including the organic compost, because we want to promote the soil health, and at the same time, we want to enhance the plant growth promotion at the same time. So here is the little attention we paid, and we created our hypothesis. And if we use the Trichoderma with organic compost, it maybe increase the nitrogen mineralization. It maybe change the fungal community or bacterial community into the soil. It maybe enhance the enzymes activity. It maybe change the microbial biomass in the soil, because we are feeding the Trichoderma at the same time when we apply organic compost as a substrate. But organic compost, we are not just using into the soil, but also giving food to the Trichoderma as a nutrient source. So here was the solution and strategies to the counter the problems and the questions. Trichoderma species and cow compost and poultry compost, because here we used already. I mean, market was available. Poultry compost and cow compost we used with the help of Trichoderma. So our overall aim was to evaluate the plant growth promoting ability of Trichoderma species, to determine the mechanism for plant growth enhancement. It will be indirect mechanisms and direct mechanisms. Indirect mechanisms mean nitrogen mineralization, who are available, nitrogen available for the plant growth, and readily, and also available from the release of enzymes, mean degradation of organic matters into the soil. And direct effects mean change in the fungal community composition, because there are many communities, fungal communities, who are responsible for the plant growth, who are responsible for the suppression of soil-borne diseases, and who will also promote plant growth. So we move to the further our materials and methods. So Trichoderma species was isolated from the soybean crop, rhizosphere soil of the soybean crop. So almost keep in mind, microbes, the host rhizosphere soil and the plant is very important when we isolate the microorganisms.
Most of the time when we isolate macroorganisms, the rhizosphere soil is, for example, wheat and rice. They are not, I mean, promoting the plant growth, because whenever you isolate the microbes, the rhizosphere soil of microbes, where the leguminous crops was grown, as leguminous crops are the nitrogen fixation crops, so most of the microbes are existing in the rhizosphere where leguminous crops was grown, are responsible for the plant growth promotion. But the I mean, like Serendipita, Basquiae, and Trichoderma, Penicillium, Aspergillus, there are many strains. And soybean, why we isolated from soybeans? Because soybean is very famous back in Japan. They are traditionally, they are eating that soybean and growing soybean after the rice. So the both organic compost was bought from the commercially. There's the company name was Kurofuji. Kurofuji is kind of organic company back in Japan. They are selling organic eggs. They are selling organic chicken, but at the same time they are selling organic compost for to the home gardening, for the horticultural gardening. So we checked the initial properties of organic compost, because initial properties are very important whenever we go for the experiment, especially incubation experiment and the pot experiment, because these properties will affect your whole results if we don't know. So here we give the name like PC mean poultry compost, CC mean cow compost, but we give the another name. OC is a oil seed cake, because there are many residues of leguminous crops residues. They mix together and they are selling commercially in the different supermarkets for home gardening. We use this OC as a positive control. So further we because we already drawn our hypothesis, our hypothesis based on the previous literatures. We used Trichoderma species in a pot experiment just to check the it's our Trichoderma species have ability to promote the plant growth or not. So here are the different four treatments were made, where four treatments were made, and two different types of plants were grown, including brassica and lettuce. So here the brassica, BR mean only brassica, BR plus TR mean brassica included with the Trichoderma species, and LC and mean lettuce, LC plus Trichoderma species. And in this part, we used only 300 gram of autoclaved soil, because we autoclave the soil first, because we do not want to affect any kind of organic material into the soil who can affect the Trichoderma and who can change the plant growth promotion. Because we want to see the just mechanism of Trichoderma species we were inoculated into the soil. So the spore suspension was made. The spore suspension was made and then applied into the parts where BR plus TR and where LC plus TR. LC mean lettuce and BR mean brassica. So I gave the name RW309, because RW309 is my strain name, because when we isolated and we submitted to the DNA data bank, and R was the name of Ryota, was my supervisor name, and W was Waleed. So 309 was my lab name, the lab room number. So we give name RW309. So for submitting the into the data bank. So at the same time, we also check the PGP traits, because every strain have a traits and have mechanisms, because plant growth promoting traits, including phosphate solubilization activity, Cedrophore production activity, oxygen production activity, and releasing of enzymes, production of enzymes activity. So we move to the further slides. Here we can see the results of that pot experiment, because the pot was put it into the growth chamber, where day and light was adjusted as compared to the day and night. And we can see the results. We can see the results only brassica compared to the brassica with Trichoderma species. The results were significantly higher compared to the only brassica was grown where Trichoderma species was not added. So if we talk about the results of lettuce, the lettuce where the Trichoderma species was added, so the results was compared to only lettuce was grown, the the results was Trichoderma species with lettuce was high compared to the only lettuce was grown. So it's mean Trichoderma species have ability to enhance the plant growth promotion as per our hypothesis. So there is a time to see the mechanisms. What kind of mechanisms? Because there is no organic material into the soil because the soil is already 2 hours autoclaved. So we checked the PGP traits as I mentioned in my previous slides. Uh, Phosphate solubilization, cytophobe production, chitnase, endogluconase is an enzyme, and phosphatase, glycosidase, and oxygen production in indole acetic acids. So positive means our strain indicate that they have ability to produce P solubilization mean ability to the strain have ability whenever we apply into the soil, it can degrade the phosphorus rated uh, compounds. So, but the strain does not have cytophobe production and chitinase production, but it showed positive sign about endogluconase and phosphatase and glycosidase and indole acetic acid. And these three traits were measured by the brewing media because we use some buffer solutions and we use PDB media to grow the Trichoderma species and incubate it for 1 week, and then we checked on spectrophotometer. So we got these results. So based on these results, we moved to our experiments, the larger experiment. So that experiment was divided into two portions. One is the portion is incubation experiment, and other experiment was the pot experiment. Because we have to check the nitrogen mineralizations and enzymes activity when we apply Trichoderma species with organic composts. So here the incubation experiment, its sign mean PCT mean Trichoderma was included with poultry compost, PC mean without Trichoderma species, CC mean I'm just trying to tell you about the my treatment because otherwise here I also mentioned CCT mean Trichoderma species with cow compost. So we incubated that part, including soil and organic compost with Trichoderma species incubated into the incubation. And we collected samples from the 0, 7, 14, 21, 28, 66, and 99 days of incubation. And we checked the nitrogen mineralization and soil enzymes activity. We measured two enzymes, phosphate latase and glycosidase enzymes. So at the same time, pot experiment was also conducted where we use different positive treatments at the same time compared to our treatment. Positive treatment was the seed cake, and another enemy, it was inorganic, that just nitrogen was applied here. So 7 treatments was conducted where the brassica plant was grown for 35 days and we placed into the incubated chamber. And after the 35 days of the maturation of brassica, we harvested that brassica and measured the plant weight and including root weight as well. And we also collected the soil sample to check to check the fungal community composition, to check the next generation sequencing, and we also collected the soil from the pot experiment. So if we move about the results of these experiments, first we will talk about the incubation experiment. If we talk about the nitrogen mineralization, so here we can see that it's indicating that nitrogen mineralization was high, where Trichoderma species was inoculated with organic compost. So it means poultry compost and cow compost with Trichoderma species, indicating that by the end of the incubation experiment, 66 days and 99 days and 100 days of the incubation experiment, nitrogen mineralization was high compared to the where Trichoderma species was not inoculated. So if we talk about the enzymes activity, phosphatase-related enzymes activity was also typically higher, but there was no significantly changes, but substitutionally was higher compared to the rest of the treatments, where Trichoderma species was inoculated with organic compost. So we can here predict that Trichoderma species have ability to degrade the organic matter and provide the nutrients to the plants. So if we talk about the glycosidase activity, at the end of the incubation, there was some substitutional results. They can change the trend compared to the rest of the treatment. So here we can see that poultry compost included with the Trichoderma species have little bit higher results compared to the rest of the results. So our incubation experiment also give evidence that Trichoderma species have ability to degrade the organic matter and provide the nutrients to the plant if we apply together at the same time with organic compost. So we move to the further results of the pot experiments. As I mentioned in the previous slide, we grow the brassica in 35 days. And here is the fresh weight of the brassica and compared to the dry weight. So if we talk about the fresh weight, we are poultry compost with cow compost with Trichoderma, poultry compost only where poultry compost with Trichoderma, there was a no significantly changes in a fresh weight. But if we check the weight about cow compost and where Trichoderma was included in that cow compost, there was a significantly difference between the plant fresh weight. So it's mean poultry compost was a little bit suitable when we apply Trichoderma instead of cow compost. So there was another, treatment. There was a positive treatment. Of course, nitrogen, synthetic, chemical fertilizers always higher compared to the bioorganic fertilizers. So we are still trying how we can maximize and how we can functions the bioorganic fertilizer compared to the inorganic, compared to the synthetic fertilizers. So if we talk about the dry shoot and root weight, because that was a fresh root and shoot weight, we put it into the oven. And we checked the another after the dry weight dramatically, the poultry compost where Trichoderma was applied, the results were significantly higher. Just because of here, maybe the water content was different. So here the water content was different after the dry biomass of the plant. So poultry cow compost and cow compost with Trichoderma almost similar results as shown in the fresh biomass. So after the plant growth, we will dig the fungal community composition as we talk about, because we have to improve the microbial community. Because as a soil macrobiologist, we have to work on the microbial community. So we just focus on the fungal community compositions. So here there are different treatments was applied during the whole experiment. So here we can see that the whole treatments are dramatically changed from each other based on PCO analysis. So here, if specifically I talk about the poultry compost with Trichoderma and cow compost without Trichoderma and without Trichoderma, so there are difference between each other. And if we talk about the synthetic chemical fertilizers, they are totally different from the organic compost where organic compost was applied. The fungal community composition was totally changed compared to the organic fertilizer, compared to the bioorganic fertilizers, and compared to the only soil as well. So if we making comparison between two treatments, cow compost, poultry compost with Trichoderma, without Trichoderma, also have substitutional difference between each other. It's mean Trichoderma is playing a very crucial role in the soil, and they are suppressing the another microbes, and they are degrading the organic materials. They are feeding by itself and also providing the nutrients to the other host microbes as well. And the results same with the cow compost with Trichoderma and cow compost without Trichoderma, also shown that Trichoderma also have changing the fungal community composition based on its mechanisms. So further, we will talk about the relative abundance of fungal communities, fungal families. So here we can see that the treatments are separately totally different from each other, but most of the treatment with Trichoderma species was applied with cow compost, and Trichoderma species applied with poultry compost are totally different. So here the two treatments change the hypochondria, fungal families, and they change the whole fungal families. And the dominant fungal families was in this treatment was Hypocristia. Hypocristia is very famous due to its plant growth promoting activity, and because most of the Trichoderma was found this in this family, because as a Trichoderma genus belongs to this Hypocristia family. Though this Hypocristia family is also very known to the promotion of plant growth and also very known to the presence of soil-borne diseases. So it's mean when we applied Trichoderma with organic compost, change the whole fungal families at the same time. So if we talk about the fungal diversity, so fungal diversity is also based on Shannon and Chow index. So poultry compost and with Trichoderma and cow compost with Trichoderma also have very different fungal diversity compared to the only cow compost and compared to the only poultry compost, because they are going to suppress the another fungal communities compared to the rest of the treatment. So based on my knowledge and based on these results or findings, I have made one conceptual diagram how Trichoderma can be effect when we apply into the soils. Because later I checked the volatile organic compounds from this strain as well, and this Trichoderma strain have ability to promote the plant growth based on the volatile organic compounds and the plate assay. So it's mean when we apply Trichoderma species with organic compost, because our soil have air and water at the same time. If some of the strains produce volatile organic compounds, definitely they will use the soil. Definitely they will use the soil air to feed the plant and to improve the nutrients. If the strain have ability to degrade the organic matter to provide the nutrients, then definitely they will use the soil water. So these Trichoderma species have ability to produce volatile organic compounds. At the same time, they have ability to produce they have ability to release the nitrogen and ability to degrade the organic matter, release more enzymes activity. So it's very important when we apply together with organic compost. It can improve the soil health at the same time, and it also enhances the plant growth promotion. So based on these findings, our one of the investor who invested on this whole study, who gave us the money for the next generation sequencing, because it's very costly. So they are now making some kind of bioorganic fertilizer, not in a larger scale, but in a smaller scale, because they were selling already poultry compost, but now they are selling bioorganic fertilizer. They are selling to the home gardening for horticultural crop, and the company name is Kuro Fuji, because they funded us. And there is some kind of conceptual diagram I try to make based on my knowledge, based on the functions of the Trichoderma species when we inoculated with the organic compost. So here are some scientific publications as well from the whole study about Trichoderma and about the whole experiment. And anyone can visit from this company by this link. So after after the completion of my PhD, I moved to the United States, pursuing my further, dreams and getting more knowledge about soil macrobiology. So the last year I joined Oklahoma State University, and the Dr. Kelly Craven, who hired me because he was also new and he wanted to build the lab. So I'm also trying to build the lab. We are not doing too much science back in Oklahoma State University because we do not have too much funds, because we already, spend it a little bit funds to build the lab. So here is my professor, Dr. Kelly Craven. He already served 15 years for the Nobel Research Institute back in Norman. So back in 2022, he moved to the Oklahoma State University. As per his words, Nobel Research Institute is not doing too much science. They are working on extensions things. So he loves always to do something about the experiment. This time we are also trying to make some kind of bioorganic fertilizers for the promotion of double wheat crop back in Oklahoma. And we already isolated the Serendipita buskii. It's very unique strain. It's a mycorrhizal strain. We isolated from the root of Bermuda grass back in Oklahoma. So we already tested that strains into the field because in my previous experiments we did not test it in the field. Here we make peat moss. We use the peat moss to grow their serendipita, and we inoculated with wheat seeds, and then we apply it into the natural environment where there are a lot of stresses and environment stress, water stress we are giving. So our strains showed very beneficial results. So based on these results, now we are also trying to make some kind of product, because every researchers have dreams to make some kind of product. His research can be a very good, very beneficial for the farmers and the society. So here is the strains as a growth media, as we are trying to making some bioorganic fertilizers, but this thing it will be definitely for a long term, is not a short term. So this is peat moss. We are using peat moss because peat moss is a very famous back in Oklahoma. I don't know about Nebraska. So peat moss is most of the gardening, most of the horticultural farmers, they are using peat moss as a substrate. So we want to use this peat moss as a bioorganic fertilizer. So here is Laila. She came from Dubai. So I'm supervising her day by day, and she's doing master. And here is Jacob is kind of managing the lab stuff, ordering some chemical and some things, because I'm not 100% aware about the how to order in the US. I was very aware when I was in Japan. So here are some achievement, because these achievement always give us the motivations. So I finished my PhD within 3 years during the COVID time. When I go to the Japan back in 2019, also COVID was also arrived with me, and he said you have to stay in the lab. You don't need to go outside. So my whole 3 year I spent it in the lab. So these are the results of 3 years. So I have published a little bit paper. Maybe these are nothing in front of you great scientists. So at the end, I would like to thankful to the USDA and NIFA, OECD, who are inviting us to sharing our research, our words to the in front of the great scientists. And at the end, I will say thank you everyone. Thank you for your patience listening my some research about some my words. So I will say thank you in English, आपका बहुत शुक्रिया in Urdu, and arigato gozaimasu in Japanese language. It's me. Thank you very much.